Hey there, folks. Okay, welcome to part 2 of the Potato Diffusion Lab. And I'm also going to take you through a little bit of the beginning of the nutrient testing lab. All right, so when we last left off, we said that your classmates had taken these two potatoes, or two chunks of potato, found the mass of each, made some observations, and then one was put in fresh water overnight, the other was put in salt water overnight. So now here's some data that you're going to write down on the second page of your blue packet, okay? This is sample data from one of the classes. So the freshwater potato on day one had a mass of 12.2 grams, and it was white, and it was very firm. It was practically crunchy like apple, okay? Because it was uncooked potato. The saltwater chunk of potato, that was 12.5 grams. It was white, and it was also firm and crunchy because they were both cut from the same potato, and we just basically cut them and took their mass. Okay. Now, on day two, after 24 hours, one was sitting in fresh water, one was sitting in salt water. Here's what we saw. The freshwater potato, the mass changed to 15.6 grams. The saltwater potato, the mass went down to 8.4 grams. And what we also saw was that the freshwater potato was still pretty firm, not mushy, still white. It had gained mass. The saltwater potato was kind of mushy and it had discolored patches, kind of brown patches. It had lost mass. Kind of interesting. So now we're going to fill in some diagrams to help explain what happened. Right here, this represents the cells of both pieces of potato. Okay, the cells started out like this. Those plant cells, kind of boxy. That's what they looked like. Now, down here, these are the cells for our potato in salt water. That potato lost mass. That means that each cell got a little bit smaller. The cells lost some mass. Whereas over here, these were the potato cells in fresh water. The cells got bigger, and that's how that chunk of potato gained mass, because the cells must have absorbed some materials. Now we're going to make a key. We're going to use little plus signs for salt and little circles for water. So let's start with the potato in salt water. Here's what was happening. The salt water had lots and lots of salt and some water as well. That was outside the potato. The potatoes had quite a bit of water inside the cytoplasm of each cell. So fill in your diagram like that, make a key, fill in the diagram, hit pause. Okay, so what happened? Well, since molecules naturally go from higher concentration inside, there's more water inside the cells, there's less water out here. There's tons and tons of salt and not as much water out here. So the water naturally flowed out of those cells, and that's why that potato lost mass, because every single cell was losing water by the process of diffusion, natural diffusion. Whereas over here, there was some natural water inside the cytoplasm of each cell, but there was 100% water outside. So there's a very high concentration of water outside, low concentration of water inside those cells. So the water molecules naturally flowed from high to low concentration. So they naturally moved into the potato cells. So that's why the potato in fresh water gained mass, because every single cell just naturally by the process of diffusion gained water molecules. They went from the water into the cytoplasm of the cell by the process of diffusion going through the cell membranes. So get your diagram up to date. And then we're just going to copy down these descriptions of the movement of the water molecules. Hit pause and copy this down. Okay, next thing we're going to do is you're going to move to the yellow packet that I gave you. The yellow packet has a paragraph at the top that's going to give you a bunch of information. You're going to use the information in that paragraph plus things that you remember from science to fill in this chart. Like, what are these chemical formulas? Where can you find these nutrients? Okay, so hit pause, use the paragraph to fill in this chart. Okay, hopefully you filled it in. Hopefully you've got the right idea that water has the formula H2O, and it's found in most foods. Glucose, maybe you got this one, maybe you didn't. Glucose is C6H12O6. That wasn't in the paragraph, but you could have looked it up somewhere else in your digestive system packet. Glucose is found in things like fruit and juice and candy. Proteins, here's an example formula for one type of protein. Okay, this many carbons, this many hydrogens, this many nitrogens, and this many oxygens, and just one sulfur. That's a big molecule. Okay, that's a macromolecule. Protein is found in meat and beans and eggs and cheese. Starch could have a formula like this. Starch is found in breads, cereal, pasta, veggies. Those are all high carbohydrate foods, so they have a lot of starch in them. Vitamins, well, vitamin C, you could have figured out the formula of vitamin C in the diagram up above, and you should have come up with C6H8O6, okay? And minerals can actually be things that are elements right off the periodic table, like iron or potassium or a few other things. Now, it turns out that the cells and tissues of all living things are made up from these nutrients listed above. That's what makes up your cells, the nutrients that you have eaten. This means that the four most common elements that make up all life are, well, one of them is nitrogen. Look at all these molecules. What are the most common elements that appear over and over in those? Well, it's carbon, hydrogen, oxygen, and nitrogen. So those are the four elements that basically make up all living things. There are lots of other elements in there, but these are the elements that are most common in living things, okay? Now, flip the page. I'm going to explain about indicators. In chemistry, an indicator is a substance that only reacts with certain molecules. And when it reacts, there is a visible color change. For example, methyl orange is this compound here. Okay. It is yellow in neutral solutions, like pure water. So methyl orange just normally is yellow. But if you add acid to methyl orange, well, then the methyl orange and the acid become the reactants in this reaction, and they make this product, which is red. Okay, so this is the indicator chemical, and when it's mixed with acid, it turns from yellow to red. The reaction proceeds if this methyl orange it mixes with acid, and they can then become reactants, and the product is methyl orange with an extra hydrogen added to it, which has a red color to it. So let's just make sure you understand that. What color does methyl orange start out as before the reaction? It starts out as yellow. What color would you expect to see if both these reactants are mixed, methyl orange plus acid? It would turn red, okay? Now, here are some more questions, making it a little more complex. Will the reaction happen if reactant 2 is missing, if there's no acid? What do you think? Well, no, the reaction can't happen if you're missing one of the reactants. So what color will you expect to see if reactant 2 is missing? It'll be yellow. So methyl orange will turn red if there's an acid present. It will stay yellow if there is no acid present. So using that information or going back to the reading, try to figure out this chart. A student tests solution X, and with methyl orange, the methyl orange is yellow. What do you think? Is the solution that they're testing an acid or neutral like water? Well, it didn't turn red, so it must be neutral like water. They're testing solution Y, and the methyl orange turns red. Is the solution acid or neutral? Well, methyl orange turns red when there's acid, so that must be an acid. And finally, they test another solution, solution Z. The methyl orange stays yellow, so that means that the solution must be a neutral solution, okay? Now, at this point, you are going to hit pause, and that is where we're ending for now. And hopefully you will be back to actually do the nutrient testing lab with us and try some indicator chemistry on your own.